Marissa Lake. I'm a freshman biology major, and today I'm here to tell you about the experiment I conducted called What's in the Water. So, the purpose of this experiment was to identify an unknown compound that's in drinking and surface water. Specifically, I identified unknown compound K. So, why is this important? This is important because we need to know if this compound has any health or environmental effects that could be potentially dangerous if it is found in the environment. I used two pieces of equipment to conduct this experiment, and I'll tell you a little bit more about them so you understand. I used the infrared spectrophotometer, or the IR spectrophotometer, and what this does is it passes infrared light through a compound to identify what functional groups are present in the unknown compound. And this is represented by different peaks on a spectra that's printed out, and each peak represents a different functional group that is present. I also used the nuclear magnetic resonance, or the NMR spectrophotometer. And what this does is it passes radio waves through the compound and identifies different hydrogen atoms that are present and the ratio between them. Not all NMR spectrophotometers identify hydrogen atoms, but the specific one I used does. And I could have additionally used a UV spectrophotometer, and what this does is it passes UV light through the compound to determine the light transmittance of a compound, but that piece of equipment was not used during this experiment. So for the procedure, the unknown compound K was placed in the IR spectrophotometer. The IR spectrophotometer is the one shown at the top, and then after it went through the IR spectrophotometer, the IR spectra was printed out. Unknown compound K was then placed in the NMR spectrophotometer, which is shown at the bottom, and then a spectra was also printed out after it went through. Once they were printed out, both spectras were compared to 15 molecules to identify what unknown compound K was. So table 1 shows the IR spectra of 15 molecules. These are the 15 molecules that unknown compound K could be. And if we ran all these 15 molecules through the infrared spectrophotometer, there would be different functional groups present at different peaks. So for example, how I found that out instead of running it through the IR spectrophotometer was looking at the structural formula. Here we have the structural formula of methanol. And as you can see, methanol has an OH functional group and a CH functional group present. So I put those in table 1, and I did that for each of the 15 molecules on the table. Table 2 shows the NMR spectra of 15 molecules, and I did a similar approach to find out the different hydrogen atoms that are present. The number of peaks on table 2 represents the number of different hydrogen atoms that are present, and the ratio of the areas represents the hydrogen atom ratio. So for example, here we have methanol again, and there are two different types of hydrogen atoms. There's a hydrogen atom connected to the oxygen, and then there's a hydrogen atom bonded to the carbon. So that would indicate there are two peaks on our spectra. And the ratio between them would be 3 to 1 because there are 3 hydrogens that are connected to the carbon and only 1 hydrogen connected to the oxygen. And I did this for each of the 15 molecules in table 2 as well. So before we look at the IR spectra for unknown compound K, we have to know how to read the spectra. So table 3 shows the characteristics of the absorption peaks that I got from the Chem 120 lab manual. And here we have the 6 functional groups that we are looking for in our IR spectra. There are more, but we are just looking for these 6. So for example, if we're looking for the OH functional group, there would be a peak on the spectra between 3,500 and 3,100. And for example, if we were looking at a peak at the bottom of the list for CBr, there would be a peak between 650 to 550. Now we can look at the results of the IR spectra. Graph 1 shows the spectra of the unknown compound K. And there are many peaks here, but the only peaks we're concerned about is the peaks that I showed previously that correspond with the 6 functional groups we are looking for. So there's a peak at 2,953.63 centimeters to the negative first, which is right here. And this peak indicates a CH single bond functional group. The next peak that we are concerned with is the peak at 771.01, which is right here, centimeters to the negative first. And this indicates a CCl functional group is present in our unknown compound K. Graph 2 shows the NMR spectra of our unknown compound K. Now, in this spectra, we see there's only 1 peak here. So if there's only 1 peak, that means there's only 1 type of hydrogen atom, and there's no ratio between the hydrogens. So now we can identify what our unknown compound K is. So first, to identify it, we have to look at the IR spectra. The unknown compound K had a CH single bond and a CCl functional group present in its IR spectra. In the molecules in Table 1 I compared it with, there were actually 3 molecules that had the same functional groups as our unknown compound K. Those molecules were 1,4-diachlorobutane, 2-chloropropane, and 1,2-diachloroethane. And since there are 3 molecules that are similar to our unknown compound K cannot yet be identified. Therefore, we have to look at the NMR spectra, which is why we had to use both the IR spectrophotometer and the NMR spectrophotometer to identify unknown compound K. So once we look at the NMR spectra of unknown compound K, we know it has 1 peak and no ratio, which means it has 1 type of hydrogen atom, and that's it. So I compared that with the molecules in Table 2 that I showed previously, and there were actually 3 molecules that had the same peaks and no ratio as unknown compound K. They were acetone, benzene, and 1,2-diachloroethane. So now we can identify unknown compound K as 1,2-diachloroethane. If you're still wondering why, it's because it has the same functional groups, which was CH single bond functional group and a CCl functional group. And it only has 1 type of hydrogen atom as shown on the NMR spectra. So now that we identified unknown compound K as 1,2-diachloroethane, why is this important? This is important because it has environmental and health effects that I found from the Agency of Toxicity from the CDC. The environmental effects I found was actually that the compound usually breaks down in water and evaporates into the air. However, if it doesn't break down and evaporate into the air, it can actually get into the nervous system and damage the kidneys, liver, and nervous system of animals who drink the water with high concentrations of this compound. It also has human effects as well. It can affect the nervous system, it can cause lung damage, and it can even cause cancer when large amounts of this compound are consumed in water. The CDC didn't exactly say what large amounts were, but we do know that if large amounts of this compound is consumed, then it is dangerous to humans and the environment. So this whole experiment was important because if this compound is found in drinking and surface water, we need to know not to drink it and how to get it out of our environment so we do not have any human health effects or environmental health effects.